How can metabolomics help understand chronic fatigue? How can metabolomics help us understand chronic fatigue? Metabolomics is the study of small molecules called metabolites in biological samples. This field can greatly enhance our understanding of chronic fatigue syndrome, also known as CFS or ME, by revealing specific metabolic abnormalities associated with the condition. By examining these metabolites, researchers can identify unique chemical signatures and disruptions in metabolic pathways that reflected biological dysfunctions in patients. Studies in metabolomics have shown that individuals with CFS-ME often exhibit a distinct hypometabolic state. This means their metabolic activity is reduced, which can be seen as a protective response to environmental stress. This hypometabolic profile is linked to mitochondrial dysfunction. Mitochondria are the energy-producing parts of our cells, and when they do not function properly, energy production decreases. This aligns with the primary symptom of profound fatigue that many CFS-ME patients experience. Metabolomics can detect changes in metabolites related to energy metabolism, including alterations in the tricarboxylic acid cycle intermediates, such as citrate and isocitrate, as well as increased levels of lactate and succinate. These findings indicate disrupted mitochondrial energy pathways and heightened oxidative stress. Moreover, metabolomics can help identify potential biomarkers. Biomarkers are measurable substances that can assist in diagnosing CFS-ME or tracking disease progression. Research has found differences in the expression of proteins involved in ATP synthesis and enzymes of the Krebs cycle in patients. These differences may serve as diagnostic indicators that show promise in sensitivity and specificity. By combining various metabolite and protein markers through advanced data analysis, researchers can enhance diagnostic accuracy, although further validation in larger patient groups is necessary. Metabolomics also aids in defining metabolic subtypes within CFS-ME populations. This reveals the diversity of metabolic disturbances among patients, which can explain why symptoms and treatment responses vary. Understanding these differences may lead to more personalized therapeutic approaches. Additionally, metabolomics highlights systemic oxidative stress and altered amino acid metabolism in CFS-ME, contributing to the complex nature of this condition. In summary, metabolomics serves as a powerful tool for uncovering the biochemical abnormalities underlying chronic fatigue syndrome. It maps metabolic disruptions, identifies biomarkers for diagnosis, and informs potential therapeutic targets. This insight supports a more objective understanding of CFS-ME, moving beyond just symptom-based diagnosis. Current scientific consensus suggests that mitochondrial dysfunction and altered energy metabolism are central features of the condition. However, while the findings from metabolomics are promising, they are still part of ongoing research and requiring further validation before being used in routine clinical practice. It is essential for patients to seek personalized medical advice from healthcare professionals for proper diagnosis and management.